Okay, yet another complication in our study of alleles in genetics. So, we can have a situation where we have a gene where one is dominant over the other. So we've seen the dominant/recessive situation. We can have a gene where we have one that is, or two, I guess, that are sort of incomplete dominance, where they both express together and we get a blending of traits. Blending. Or we can have a situation where they have two different variations, but instead of a blending, we call this co-dominance. Okay, I should have written dominance up here, incomplete dominance. This one's called co-dominance, and we get the blotchy. We get both traits expressed. Okay, so the blending gives us a third option, a third trait, like blue and red making purple, whereas this one would be blue and red making blotchy blue and red. Okay, now you'll notice that every time here that we've done this, we have used two different alleles. A gene that comes in two different forms. Now, you only get two copies of a gene because in your cells you have the two homologous chromosomes, right, that each carry one of these alleles. But that doesn't mean that there can only be two alleles in existence. It's possible to have what we call multiple alleles. So the next situation is what happens when there are more than two alleles in a population that are possible. And the best way to understand this is through an example. And the example that we'll use is human blood types. Human blood types are also genetic. Now, I'm going to have to explain a little bit how blood types work and what that actually means. You may know what the different blood types are. You know that there's type A, there's type B, there's type O, and then there's also type AB. And you might be wondering what exactly that means. So I'm going to try to explain it. On your blood cells, on every cell, for instance, there's a cell membrane. And the cell membrane, let me just move down here. The cell membrane is actually a very complex structure, and we're going to learn a lot of this later on in the course. But the cell membrane is like a layer of molecules. And on top of that layer of molecules, there are other molecules that stick out. And these molecules that stick all out of the membrane are, they serve many different functions. Some of them help hold cells together. Some of them are like license plates that identify the cell and where it belongs. This is how your immune system can tell if a bacteria cell belongs to you or not. But another thing these things do is they have signaling situations where other molecules can come along. Here's another molecule, and they can bind or fit into these. It's almost like a lock and a key, right? And if the molecule can fit in the right place, what it does is it changes the shape of the membrane when it fits in there, and it affects how the cell behaves. So molecules that are bumping into the cell from the outside, if they're just the right shape, they can activate these, what we call receptor sites, and change the way a cell behaves. And there are many different kinds and many different options here, but the one that's important to us is this human blood type. Okay? And so we have a name for this. We call it an antigen. So the antigen is essentially a molecule on the cell membrane. It's one of these molecules that I've been talking about. It's one of these signaling molecules that's sticking out of your blood cells. And there are a couple different types. So we call one antigen A, and we call the other one antigen B. There's actually only two. All right. So what does this mean? Well, it means that if you have a blood cell and this is the membrane. And on that membrane, you have these little antigen A molecules all attached to your membrane. Then you are what we call type A. Now, how would you get this happening? What kind of genotype must you have? Well, you're going to have two genes. We use an I for the allele name. I'm not sure why, to be honest. I has nothing to do with anything, but maybe that's just so we don't get confused. So I is the typical blood type allele, and then we put the A up here for type A. So your genotype here would be type A because all you have is A antigens on your blood cells, and you have no genes for anything else. The other option could be that you have blood cells that have this little antigen B on them instead, because you have different genes, right? If you have this, then you have type B blood. And what this means is your genotype is alleles that carry this version of the gene. So these are your alleles. That's type B blood. But you can also have an allele that doesn't make either one. So the third option is an allele. Okay, so I'm going to write this down. There is a third form of, that says form, of allele. No antigen. It doesn't make A, and it doesn't make B. So, here's the interesting thing. This antigen is going to be a little I. And here's the reason why. Okay, so, well, first of all, let me just show you what would happen. You'd have a blood cell, and you would have none of these little molecules around the outside, and your genotype would be little I, little I. Now, why did I switch to little I instead of using the big capital I? Because there's three alleles, they all interact differently. These two alleles, IA and IB, are more of a codominant situation. So if they're present, you get both A and B together. But both of these are dominant over the one that makes nothing, little I. So we have two of the options that are codominant with each other, and one option which is recessive to both of the others. So we've seen these two blood types, type A and type B, right? So what I'm going to do is just, and then we've seen this third blood type, which has no antigens. This one has a name. I should have written it down. It's called type O. If you're type O, O means you have no antigens on your blood cells. And then the other option could be if you had one of each of the codominant alleles. You could have an IA and an IB, and guess what? Because they're codominant, your blood cell would have both A and B antigens, A and B. So you get a little bit of both. So this is called type AB. So just to summarize, okay, if you have IA, so you got one of those from your mom and one from your dad, right, in a sperm and an egg cell, you would be type A blood. And if you got a B instead from mom and a B from dad, you would be type B. But there's another way you can be type A and type B. You can also be type A if you have this situation. You got the little I from mom, let's say, and the type A from dad. This is also type A because the allele for A is dominant over the little I. And that would be called a heterozygous type A. This is a homozygous type A on the other side. And you can also be a type B if you had this situation. because the B allele is also dominant over the little I. So there's two ways to be type A and two ways to be type B. But there's only one way to have both. And then you would, this because they're co-dominant, you'd get a little bit of each, and we call this type AB with both of them. And then the last combination would be if you got two of the little i's, one from mom, one from dad, you'd have no antigens, and you would be type O. And that's how it would basically work, right? There's only one way to be type AB and only one way to be type O. But there are two ways to be type A or type B because of the dominance of the A and B alleles over the little one, the little I. Okay, so now you could do all kinds of crosses with this multiple allele to figure out, blood types. So let's just do a typical cross just for fun. Let's say we have a mom who is type A. That's her phenotype. But her genotype is this. She carries this little recessive allele for no antigens. So her blood cells are going to have A antigens on them because of that dominant allele, right? And she will look like she's type A from a phenotypic point of view, but she's heterozygous. And then let's take a father who happens to be type AB. That means his genotype is an A and a B. Well, now we can figure out what the probabilities would be of the children's blood types by doing the cross. So father, IA, IB, mother, IA, little I. And so what we would get here is a homozygous type A heterozygous type A, just like mom. We'd get an AB, just like dad. And this is interesting. We would get a heterozygous type B, right? Which is not mom or dad. Whoops, didn't mean to make that mark on there. Mom or dad were not type B, but somehow type B has shown up. So let's just do the percentages here. Right here we see that there's 1 out of 4, which are homozygous, but their phenotype is type A. And then we have a 1 out of 4 heterozygous, but that's also type A. So we could put those together, right? If we put those together, we could say that there's a 2 in 4 chance of being type A, because they're different genotypes but the same phenotype. They both will have A's on their blood cells. And then if we go back up here, we can see that there is 1 out of 4 for heterozygous, but this time it's the 2 codominant ones. So it's A and B, so that would be type AB. And then lastly, we have this situation right here where we have a type B, 1 in 4, heterozygous, mind you, but that B allele will block the tiny little I that's for no antigens, and this will be a type B. So you can get a type B out of this, even though mom and dad were not. And you can easily see why, because dad carries the B allele right there. And if his sperm happens to get that B allele through meiosis, then that's the one that would combine with one of these from the mother, and it would result in these two, the green and the yellow, and you can get a type A or a type B. And you can do all kinds of Punnett square combinations and get different matches from that. But the idea here is to just understand the idea that what we have is a multiple allele system where we have 3 alleles. And to also understand that sometimes among the multiple alleles, some could be codominant, some could be dominant, some could be recessive, like this one. Right? This is a recessive one. These are dominant, but together they are codominant. So all of the things we've learned so far can be thrown into the mix to complicate the issue. And this is how we see so many variations in traits. Some traits are controlled by not 1, 2, 3 alleles, but maybe 4, 5, or 6 possible alleles. And they get very complicated. So this is what our modern genetics